All right, good afternoon. I guess we can start as folks are joining in. My name is Sanjay Jain from Washington University in St. Louis, and I'm part of the HubMap Consortium and the Kidney Precision Medicine Project, also known as KPMP. And I'll be moderating this session. And just to give you a brief background, that there are two other sessions based on tissue. One is on tissue collections, different types of tissue collections by sites, and the other one is characterizations. And this one is focused on quality control and assessment for different technologies. And hopefully you are in the right session. So if I could have the active participants, which we would like several of you to turn on the video so we know who the active participants are, that would be great. And we would like the passive participants to mute themselves. So could I have some of the active participants introduce themselves briefly as well? I see we are a nice small group of about 9 or 10 folks over here, so we can get a lot of stuff done during the session. So yeah, please go ahead. I see Alexander, I think you may be in mute.
Yes, all right. So my name is Alexander Denadai. I'm from KU11. And I'm working on the Human Gut Cell Atlas consortium. And we're running a bunch of 10x, and we start some spatial transcriptomics as well.
Okay, thank you.
You're welcome.
I see Gloria. Hi, Gloria.
Hi, Sanjay. It's hard to know if we're going to be active participants or not in any group, but I'll introduce myself. I'm from the University of Rochester, working on both the lung map and the HubMap program. Very interested to hear this session.
Thank you. All right, and anybody else who would like to be an active participant? Feel free to jump in, as the discussion goes. We're pretty here, informal in a way to have a really productive meeting here. So, and then we would like a couple of volunteers to take notes. So if somebody could volunteer, anybody in this, I think that would be we really appreciate that, please. So. So I guess everybody should be able to also open the Google Doc. That's the link in the KI website, which has this doc with some guiding questions for this group to address. And if the scribes could just take notes as the discussion is going on. So could we have some volunteers for that, please?
Sanjay, I'm putting a link to that right in your chat here in case it's helpful to anybody.
Okay, great, thanks.
And I've also put my email address in there in case you need anything. It sounds like you've got the process under control, so I'm going to go visit some of the other rooms and I'll be back momentarily to check in with you.
All right, thanks, Robert.
Thank you.
All right, so, well, I guess I'm going to follow the guidelines about see if we can volunteer some folks to scribe. It's really just taking notes, and anybody can feel free to put in the sheets so we don't miss anything. We just need somebody to type in some notes or key questions or problems that we identify during this discussion while some of the folks are talking. So I'm just going to the participant list. Let's see. Yeah, I thought I heard something. Okay, so let's see. Bradley and Ellen, could we have your services for scribes, please? or anyone else? Okay. How about, I think Bradley, maybe shifting. So how about Ellen? You could unmute yourself so we can hear. You know. All right. We have Fiona. We have Jennifer. Okay. Thanks, Jennifer, for volunteering. Really appreciate that. All right, so let's start and with the discussion. So as you all heard, maybe in some of the morning sessions, at least the session I was in, that one of the key things that keeps on coming is, especially for a lot of these single cell omics and imaging technologies, is their quality of data are really dependent on how tissue is procured. So we'd like to hear some thoughts from all the participants across different consortia, is how are they doing quality control and assessment, and what are the challenges they're facing in each of the discussion prompts or from their own experience. It could be from beginning from patient selection to metadata or pre-analytical parameters or histopathology and so forth. So let's get the discussion rolling on that and see what people are struggling with or what their methods are. Why don't we start with Alexander and Gloria, and then we can open the topic to others.
Sure, I can start on that. At least to give a quick overview of the tissues we're dealing with and how we're trying to get the best out of it. So as I mentioned before, we're working on the Human Gut Cell Atlas, specifically funded by the Helmsley Trust. So we are working mostly on the ileum and ascending column as well. And we are interested in two particular cell types, macrophages and as well as the epithelium, particularly the secretory lineage. So for the patient selection, so we're actually it's more, we're quite pragmatic. For the patient selection, we try to figure out which subtypes of patients would give us samples, the best samples. So for instance, for macrophages, we're working mostly with resections from patients that undergone colectomies or other procedures. And then in the column itself, we're mostly working with biopsies. One thing that for the inclusion exclusion criteria, of course, there are many details involved, but we try to have as much information from these patients to determine if they can be considered or not a healthy control, let's say. So we have endoscopy data from these patients. We have calprotectin measurements, C protein as well. So these are some elements that we're trying to get very well established to define what whether a patient can be considered a health control or not, at least the tissue that we are targeting. What else I could say about it is that another thing that it was quite of a challenge for us as well. We realized by running some preliminary experiments that the freshness of the tissue is really important, especially for us that we are running 10x single cell RNA sequencing, spatial transcriptomics by different methods. So it was quite of a challenge to exchange with the surgeons or the endoscopists to get this tissue as fresh as possible. So we managed to get a pipeline that we can have this tissue being processed within half an hour from the time the sample drops. So these are some of the first considerations I would do about it. Of course, besides the metadata, we also have histopathology of every single sample that is included in the study. So in general lines, these are the main measures that we took into consideration to have the samples be included in our studies.
Okay, thanks. Yeah. So Gloria, would you like to go ahead? Maybe on mute.
Unmute, yes, there. Yes, so we face a challenge with the lung samples in that, especially for our pediatric samples, biopsy is very uncommon. And so we've taken the approach of working through the transplant network and minimizing the warm ischemic time as much as possible. The point that was just made about freshness being important is not lost on us. But especially for our pediatric samples, we do the best that we can there. One of the other things that we've noticed is that most any lung that's on a ventilator for any period of time has inflammatory cells in the alveolar space, not so much in the tissue, but in the alveolar space and somewhat in the tissue. And so they're in talking about quality, and we do look at viability of the cells that we isolate. We look at RNA integrity. We look at numbers of cells that we're able to access, etc. So what we've come to is that the quality and the normality of the tissue that is required may be different depending on what the analysis is that is to be done, right? So what I'm trying to say there is the health of the tissue, the health of the cells, that's clearly very important. But whether there is some inflammation there or not, whether there is some in the lung, we get hemorrhage not infrequently in certain areas. Depending on the analysis to be done, the sample may or may not be appropriate, depending on that underlying abnormality. So I guess that we're trying to, how might we questions. I think one of the important things maybe is to figure out what are the important QC elements for any particular analysis or any particular question, right? If you're looking for inflammatory cells, maybe you want the lung that's more inflamed. If you're looking for a pristine, normal lung, then that's not a sample you would choose. We do try to get, so the metadata, we try to acquire, these are all human subjects. They all have, they all carry baggage. So acquiring as much metadata as we can in terms of their prior exposure. So again, how might we determine what the useful metadata is, what the necessary metadata is, and trying to convince those that provide the samples that it's important that we get the metadata, their exposure history, as well as their current disease history.
Yeah, thanks, Gloria. I can just tell briefly about the kidneys. So we are studying the kidneys and the lower urinary tract, and our specimens are from deceased donors as well as from surgical specimens. And the issues that we certainly face, that's echoed, and it's a challenge selecting the patient. So almost sort of quote unquote reference atlas, try to screen the patient's chart for comorbidities, and especially if they have underlying already known kidney disease or kidney function. So I guess for maybe many organs, that is a maybe criteria if you're making a reference for the organ in question. Maybe some functional parameters in screening would help, or underlying chronic disease of those organs would help. So we do those, and we also look at the the freshness, the time factor, I guess, which is important. And I think Gloria mentioned about ischemia times and so forth. So track some of those time factors. And one of the challenges, as Alexander mentioned, is for us for kidney resection as well, is coordinating with the surgical team and to keep in constant touch with the OR to tell us, let us give a heads up when the specimen is coming out, so we can actually monitor the time of warm and cold ischemia and transport, and how long do the pathologists take to triage the specimen before we can use it, for research purposes. So at least for the diagnostic tissues, that is a sort of a concern, and can affect quality, and some of those, I think, things are important to identify, at least in our patient selection. Anybody else in the group who have experience there, and what challenges they face in terms of patient selection or organs there? We can pause and listen from the group. Anybody, feel free to unmute yourself and chime in here. Okay, so maybe then one of the questions I'm hearing is, like, how might we sort of maybe standardize these patient-based parameters, whether it's radiological evaluation or other clinical functional parameters, to track with as a quality control. Could be maybe one of the challenges that maybe are important for the outcome of the data. Is that an important problem? What the group thinks? Track. Okay. So these are the metadata. So we would like to hear about, sort of briefly touched about this, is what are the specific, parameters or that are critical for the metadata affecting the tissue quality. So let's hear from the group about that. So, let's start. We can start with Alexander, and then Gloria, and then the group.
For the metadata itself, I don't know exactly from where to start regarding that, but we have 2 trial nurses that take care of this in our consortium. So actually, the metadata, we have access to the whole information related to a given patient or a health volunteer that joins the study. I think this is end up being quite dependent on the organ itself that each one is working with. Of course, there are general things that are important to everybody, like age, sex, and many other aspects. But other than that, yeah, some clinical parameters regarding general health, such as C-reactive protein, for instance. We also collect blood from these patients. But other than that, I'm not sure I'm in the best position to really contribute on this topic.
How do you decide, like, what kind of metadata do you need, as a measure for your gut cells, or maybe who other folks who are doing gut work?
To be honest, we are still figuring out about these things. As I said, we have everything that is possible to have from each subject, but we are still trying to figure out what should be included in this metadata once depositing the let's say the results of our experiment, for instance. So this is something that we still really don't know. We are still trying to figure out what is important to be there. So yeah, perhaps this is the right occasion to start discussing this, for instance. Besides the obvious topics like age, sex, and other general health parameters, what is really important to be there as a whole for the development of the Human Cell Atlas.
Gloria, your thoughts about metadata, what should be collected and how you decide?
I'm the one that's typing in there the how might we. So I agree, standardizing the clinical metadata that is needed and that will be helpful in analysis. And then something Alexander said was, does it vary by tissue type? It likely does, right? And the physiologic markers that you mentioned. You know, just suggesting that maybe small groups of investigators who, or physician scientists who are familiar with a particular organ, familiar with what exposures might be important, et cetera, would be good to get together to try to standardize the metadata that's collected, and agree that some of them are pretty obvious. Some of them will be really specific to the organ, specific to the, even specific to the age, right?
Right, yeah, absolutely. Yeah, yeah. So anybody else in the group who has not spoken would like to unmute and give us their thoughts about what essential components they think are for metadata collection, or how they do it, you know, how they solve this challenge? Okay. Yeah, so I think for kidney also, you know, we have this same challenge, is where to stop, because, you know, there's so much data out there. We have to make our best judgment, and we sort of focus on only kidney parameters, you know, but I'm sure we are blindsided by things which may be important, such as some of the respiratory or lung function measures, which may be affecting the kidney, but we are usually not really paying attention to them. So I think, you know, we are certainly, when we start thinking about awareness of other organs, brain, lung, heart, you know, we probably maybe should pay attention to those as well, because we don't know what is affecting in other organs' health to our own organs profile. So it's something I think is a blind spot, at least when we are tunnel visioned in our own organ system. But the other side of that is of course, the resources that are needed. But if maybe this is a problem that maybe the group can collectively maybe figure out as a challenge to address, what could be the minimal essential parameters from all the vital organs that maybe we should pay attention to, you know, and maybe an interesting one, like how might we, coalesce together to tackle or identify maybe from each organ what are the most important vital functional characteristics that could affect expression in other organ systems or so forth. So yeah, I see this as a challenge where at least we are sort of like victims of, yeah.
I think that's really important, Sanjay, and part of it too is educating investigators to know what to think about, right? To know what might be important. So I'll give you an example. For us in the lung, we know that the lung cells respond to 100% oxygen, right? It's toxic to the epithelial cells, so do I gather every blood gas? Do I try to get them, to download for me every oxygen saturation for the duration of the time in the hospital? Probably not, but somehow, how do we get a handle on that? And then just as you were saying, so I know that that's important for me in the lung, but if a patient has been kept with hyperoxic for some period of time or hypoxic for a period of time, that's going to change how your cells in the kidneys look.
That's good.
And so, yeah, how do we capture those kinds of, I'll call them exposures? How do we put them into any kind of analysis, you know, without taxing our facilities, taxing
Right.
the ability to gather that data and analyze it? Yeah.
Right. Yeah. Yeah. So yeah, no, I think absolutely it's sort of critical to identify those. Anybody else have thoughts? Yeah, feel free to, you know, unmute and, you know, comment, you know, so we can capture as many ideas and challenges that people are facing so we can address those. Okay. All right, how about some of these pre-analytical parameters, you know, for your assays, you know, during processing and so forth. So Alexander, how, what kind of challenges or what things do you track in your assays, you know, which are critical for your quality assessment and control?
So we are quite in the beginning yet, but I would say that as Laura mentioned it as well early on, I think that one important aspect during this whole process is cell viability. This is something that we had in mind all the time from different sorts of samples, resections, biopsies, different cell types as well. We realized that the requirements or stringents are quite different depending on the cells we're targeting actually. Realize that for the epithelium, for instance, timing and temperature are really important to keep viability. For macrophages, not that stringent, for instance. So this is something that we measure multiple times before loading the cells into a 10x platform, for instance. So yeah, this is something that is really important for us.
And how do you assess viability? What kind of assay do you use?
So we have a cell contest, and also a contest Luna as well. So we use both of these strategies. For the early stages, we use a cell contest, and right before loading the cells to the 10x chromium, then we use the Luna with the fluorescence kit. It's a bit more accurate. So yeah, we have these two strategies that we use in many different steps during the cell isolation until the very end or loading of the cells into the chromium.
So is there a cutoff you use where viability, you know, that you use, where you say it's a go or no go for your particular gut cell types?
It's around 85%. If it is below that, we don't go on with the loading of the cells. Of course, we have many steps to enrich viable cells as well. We have a sorting in some of our experimental designs to enrich for viable cells. But in the very end, let's say 85% is our cutoff. Below that, we stop.
And so do you use any other, you know, attributes like does RNA quality correlate, or, you know, somebody mentioned RIN values, or, you know, DV200s or other things that you use for quality control, or they don't matter?
Later on, yeah. Before the library preparation and
Okay.
these steps, yeah.
Okay. Gloria, would you like to comment on that? I think, Gloria, you're on mute. Okay, I think Gloria may be on mute. Anybody in the group? I see there are folks who do single cell technologies there too. So Edgar, do you want to comment? If you are on, you unmute on your single cells. How would you use for your pre-analytical parameters?
Yeah,
Can you hear me? Yeah, so what we are doing is 37-degree dissociation for 12 minutes. So this is, and there is no sorting involved. So we're fast, but there is the viability goes, of course, down. And I have the experience that we have with the precious material. We are using biopsies, kidney biopsies, and there is no way to sort them to don't waste our material. So we have also counters, but there will be no relation, whatever viability. So we have a viability between 10 and 70%, but this is not consistent. So even if we have 10% viability and have enough cells, we load them on the 10x and we'll get a lot of QC. After QC, after a cutoff, we get enough cells from these samples. So there's not, for us, it's not reliable to rely on the counters measures. So we use all the cells we can get and put them on the 10x, even if it's more than 10,000 cells. Usually we'll put all the cells on the 10x genomics and see what cells make it through our cutoffs.
Yeah, thanks, Edgar. So for both of you and the group, I guess, how about like the amount of time it takes to process that tissue? Is there a go, no go, or some parameters that you have identified, in terms of tissue from harvest to processing or putting in preservation, that there's a critical time where your tissue is not usable or have good quality?
I could comment on that. This is not something we have validated. We just try to make things happen as quick as possible. So I didn't mention it before. Time of single cell dissociation was something that we took into account as well. So for the epithelium itself, we tried 10 different enzymatic preparations. Ideally, we would like to go with a cold method, a cold active protease. We tried that. We had very good cellular preparations with it. The problem is that the surface antigens were all digested.
Mm-hmm.
So since we have some multi-omics experiments previewed later on in the project, and in which we will have to select these cells, enrich these cells a bit more. So this was a no-go. So then we had to move to 37 degrees, and then we found an enzymatic formulation that was giving us a good prep in 5 minutes. So in general, protocol takes from the dissociation of cells, staining, until sorting, it's, let's say, between 2 and 3 hours. Not because this has been validated. It's just that we think that the quicker, the better. So this is our strategy in this regard.
Okay. Others, Gloria?
Yeah, I'll try to keep myself unmuted when I'm talking. I'll just comment from the repository point of view. So much of our funding is just as a repository. And a challenge is getting the materials, although the viability, for example, might be quite good in our lab, to send that to another lab and see it through their process. It can be challenging. Things happen, whether it's in shipment or it's in arrival in the other lab. And so communication, again, of protocols and handling of material can be challenging and can really affect the outcome.
Okay. Yeah, thanks. Edgar?
Yeah, I can comment on the cold active protease, which I also tried for a period of time to reduce the stress or to avoid stress reactions of the cells. And I can tell that I've done 15 biopsies with the cold active protease, and the same from the same biopsy, another section of the biopsy done with our 37-degree Liberase dissociation method. And I can tell that we have also the viability looks great with cold active protease, about more than 50% all the time, looks perfect. But after 10x genomics runs, we have actually a magnitude 10 times more cells which have enough genes per cell when we use the 37 degrees. So we gave up on the cold active protease. It's really acting on at 4 degrees, destroys the cells. Also, they look good afterwards. But after 10x genomics, there's a huge difference with a 37-degree protocol, which we now use. So we gave up on the cold active protease.
Yeah, yeah, sure, go ahead.
Sorry, no, I was just going to say that it's reassuring to know that we made the right decision to give up about the cold active protease. It looked promising in the beginning, but yeah, for different reasons we stopped with it. So it's good to know that we were having artifacts with it. So yeah.
Okay, let's quickly go to assessment of your tissue by quality by pathology. Do you guys use that for your tissue quality, the quality of tissue you're getting, and what do you use?
I'll speak for us again. We have two pathologists, one primary pathologist who reviews every sample. She chooses the blocks kind of randomly around the organ and reviews H&Es. If she needs more, we send her more. The downside is it's not always directly next to the sample that we send for single nuclear or that we've dissociated for single cell. So that is still a little bit of a challenge to have the histopathology in the same area that the analysis is happening. But it does help to identify disease where we didn't expect it, to identify autolysis, to identify inflammatory areas that we might want to stay away from.
Okay. Yeah, I think that's very important. And Alexander, how about your group? Do you use that as a quality assessment and control?
Yeah, we do it as a routine. It's a bit that, of course, since we work with fresh tissues, by the time we have the histopathology report, the samples are already processed. So it's not something that is driving the tissue selection itself, because it's just how our pipeline is operating. But we do for every single tissue or patient, we do a routine histopathology analysis, but this is done by the hospital itself, by the pathology section of the hospital as a routine. So this is how we are dealing with this.
So do you have a set of parameters or minimum parameters that they check for and inform you on, so that can help you in your interpretation of your results? Or is that something like something to still be worked out?
It's quite general for us. It's mostly the presence or absence or the degree of inflammation present in the tissue.
Okay.
I mean, besides other architectural parameters of the tissue, but mostly inflammation.
Okay. So is this maybe an area of like how might we want to, across different organs, understand what are the minimum histopathological evaluation or composition of tissue should be, so we can correlate that as a QC measure with the omics analysis, I guess, something. Yeah, because you use for inflammation, we use for checking the integrity of the tissue, autolysis, necrosis areas, and whether the composition, because kidney is quite complex, the heterogeneity of what we actually have in our tissue and so forth. And I think Gloria is also, their pathologists are also marking some of this, right? So, Anybody else in the group who have thoughts about this regarding histopathology or other quality control measures in your tissue? How about storage? Do we have standards of how tissue or should be stored if you're going to use for later time points? Like, how do you store? Have you gone back and reused the tissue and so forth? Does it work in your case? Or I think somebody said they've been shipping. So are there set criteria for that? Especially in a cross consortia setting, if we are going to be trading tissue and derivatives, you know, across sites.
So in our case, for all the 10x experiments and multiomics, we use always fresh samples. So we don't have experience with storage, but we do have the spatial techniques. So we have the spatial transcriptomics being done in Sweden by the team of Joachim Landberg, and also sickfish, marefish doing here in K11 itself in Belgium. So in this case, we do have tissue storage as a routine. So what we're going to do, we're mostly following the general guidelines. So once we harvest a resected material or biopsies, we include it right away in OCT. And then we keep it at -80, but we don't fix the tissue previously. It's really the fresh tissue that is frozen. And as far as I know, this is the gold standard right now for spatial techniques.
Okay. How about anybody else? Gloria or Edgar or anybody else, how they handle storage and shipping?
Yeah, here's Michigan. We are using only frozen materials, so that's the only way between the consortia in the KPMP to send from the recruitment sites samples over to the tissue and irrigation sites. So we cryostore that with in cryostore, which is basically a 10% DMSO freezing of small tissue material like a biopsy. So you can freeze cells, but you can also freeze small tissue pieces. And our biopsy is about 10 to 15 millimeters in length. So this works for dissociation afterwards in 10x genomics.
Okay. Yeah, for us, for the single nucleus, we use fresh frozen OCTs, and if they are frozen, kept in -80, we have been able to come after like even 5-6 months and use that material for single nucleus sequencing without really losing much in terms of our sensitivity and clusters. Okay, anybody else have any comments? I see that we are due for a break for 15 minutes. So please feel free either to, you can type in also in the Google Docs in case we missed and something you wanted to say but were not able to. Please feel free to put it in the Google Doc, and we'll reconvene back, I guess, in 15 minutes and then come up with some possible potential collaborative effort topics. Okay, we'll see you soon. Thanks. Yeah.
Thanks, everybody.
Okay, thank you.
Hi, Sanjay.
Thank you, Jennifer, for being so diligent, taking notes frantically. Great. And anybody else who has been doing that too. Thanks. So just 15 minutes ago, we were filling and discussing some of the topics for the quality control that some of you were actively participating in. And there was a Google Doc in which I believe Jennifer and a few others were typing in how might we kind of questions as challenges were being recognized. So in this second part of the breakout, what we need to do is further crystallize on maybe 4 or 5 of some of these challenges that we can address at a across the consortia kind of level, you know, that are challenges faced by multiple folks, but we need to engage a wider community to address these and see where we can standardize some of these questions. So, so I think we can start. And since this is a small group, everybody is an active participant. So feel free to chime in and identify things which we believe should be some of the top priority items for quality control of tissue procurement and processing kind of questions, which certainly impact the downstream analysis. So, yeah.
Sanjay, before you get started, would it be useful if I gave an overview of kind of the goal for the
yeah.
output?
Yeah, please go ahead. Thanks. Yeah, sorry. Yeah.
Let me share my screen here. And you guys are 10C, correct? Yeah. So what you've done here is you've identified some good how might we challenges, all of these. And so our hope is that you would grab one at a time, or maybe a cluster if you feel like you can address them at the same time. And then scroll down here to the blue box. Let me put this on one page just to make it easy. Give that a title. Cut and paste in here the 2 challenges that you're, as many as you're feeling, the 1 or 2 or 3 that you're dealing with. List your recommendations for what you think should be done. Just write your names here, who's moderating, who's taking notes, and email contact. That way, if one of the organizers has a question and wants to follow up on this, they know who to follow up with. Any expertise you feel is needed to kind of chime in here, and then if there's any kind of next step that you've set. And if you're going to tackle more than one challenge, and most groups are, you just kind of copy and paste this over and do another one. So you just kind of do them one at a time. And then that's what we're going to come through and collect and put in the output gallery for the end of the day. Is that fairly straightforward?
Yeah, thanks, Rose. Yeah.
Excellent. Good.
So are we, can you give us the time frame for this session?
Yeah, absolutely. So it's 2:55 East Coast now. Plenary is going to convene at 4:05. So you have about an hour and 10 minutes total to try to get as many of these as you feel you want to get done. And then that main Zoom button that we've been hitting all day, that's where you're going to go at 4:05.
Okay. All right. Thanks.
Thank you, guys.
All right. Okay, let's get started. So I guess we need somebody to maybe type in the blue box. So could we get a volunteer to type in the blue box? Somebody's trying to speak, I can't hear, sorry.
I typed in the green box. It would be better if somebody else typed in the blue box.
Okay. Okay, I'll try to type in as we are discussing. So in the blue box, yeah, we have plenty of time. So, okay, let's go with the discussion from the how might we. So, go ahead. Yeah, you want to start? Maybe you can start with Alexander, and then people can chime in regarding the challenges which we think we should tackle at across the consortium level. Oh, somebody's audio is not working well.
So,
okay. Yeah.
At least in my opinion, I would say that one thing that is perhaps of broader interest and to be interesting to tackle, as mentioned before, is to define what are the minimum information regarding metadata that we should have, either both as in general as well as tissue specific. So I think this, at least in my opinion, would be something interesting to work on.
So you mean like what is the minimum metadata needed for clinical parameters as well as tissue procurement parameters?
Yeah, I think this is a burning question for most of us. I think it'd be of interest to a lot of people.
Okay. Okay, how about others? Any thoughts on that? Gloria, do you have any things from the how might we sessions, which something which we should tackle?
So the sharing of protocols, I think this comes up on some of the other sessions too, but sharing of protocols and successes and failures, as I wrote it there. So hearing somebody else say, you know, that cold active processing was not working for them just validated, I think, Alexander's findings too. So it would be helpful to know not only what has been working maybe, but what has failed or why it fails and why it fails. So yeah, places to share our, not just the end data and analysis, but maybe some of the QC work that goes before.
Okay. All right. How about others? What are other aspects that can be solved across consortium? So I guess one of the, at least for us, is that, you know, how do we really assess the quality of tissue once we get it, you know? So what should we, across different consortia, like how do we know what we're getting is good, you know? Some of the things we talked about, but I believe looks like different folks have different ways they are measuring quality of the tissue. And it may be an important exercise across consortia to see what different ways people have in their bucket to measure the quality of the tissue. Some folks are using histopathology, some are using RNA, some are using, you know, some other means, I guess, you know. You know, maybe it'll be good to understand the quality control of tissue quality across the sites, and then see where we can standardize.
So this is Rob. I guess that gets into the whole issue of, are there particular analytes that are quite stable, medium stable, not so stable under various conditions? And do some of them need to be spiked in, or are there exogenous ones that you could follow?
Yeah, that's a good point too, to come up with strategies to gauge the quality of the tissue or your assay in the beginning.
Well, the ones I was talking about were more for the pre-analytic processing, but yeah, I guess it does also, might be helpful for the tissue itself. So some of this also gets the issue of, how do you tell signal from noise, or signal from cold ischemia, or signal from poor processing? Are there certain gene subsets that get upregulated? There are, but which are the best ones to follow? And how do you tell that from the underlying biology that you're trying to find?
So a sort of a cross consortia wide list of artifact signatures, I guess, you know.
Good, I like it.
Okay.
Yeah, yes, our artifacts we have known.
Okay. All right. What else from our list? So I'm just typing in these in the green box of breakout session part 2. And then we can select from these for the blue box activity, you know. So I think, Alexander, you were mentioning about inflammatory cells and so forth. And I think, Gloria, you also mentioned, you know, that in your lung specimen, there were the way you harvest could affect or invoke an inflammatory response in the lung. So, I mean, how do you guys both think about handling that, across, because everybody will have some sort of immune cells in their, single cell outputs. So how should we tackle that? I mean, how would you know what inflammatory cells are being invoked by processing step versus what are really, part of the biological process?
Yeah, I'd say it's not just the processing, but the events that have happened prior to recovery of the tissue. Yeah, I think that is a challenge until we have built large enough databases. Until we've looked at enough to know kind of what the signature of that ventilation effect is, the oxygen effect is. I think personally, it's hard to know right now. So in my estimation, what's really important is that as we use these tissues, as we put out the databases of analysis, that the metadata be included, right? So that we know where a particular sample came from, if you will, right? So then we have the opportunity to go back and say, well, this one was different because the person was on the ventilator for 10 days versus 1 day. That sort of thing.
Mm-hmm. Okay, so that can be sort of within the framework of the metadata parameters, I guess. Yeah.
Correct, and that's going to take a cross-consortia involvement because none of us will have enough samples until at least the cost comes down, to be able to say too much by ourselves.
Go ahead.
Yeah, I completely agree with Gloria. I think it's too early to have these answers. So it's more, right now is more a matter of registering properly all these observations as metadata and see how gene expression correlates with inflammation or hypoxia, and try to figure out in the future how to go on. But yeah, at least from my perspective as well, we still don't have answers for these questions. We are not able to set cutoffs yet for, let's say, microinflammation in the tissue. So we're trying to be stringent, but also get some hands-on experience and processing some material to realize about these things.
Trying to be as transparent as we can.
Yeah.
About the samples. And it's not to say that there isn't work gone before. Stephen is on. I am sure that there is biorepository experience that would give us, a better place to start than some of us are aware of.
So do you think any model organism studies would help in this sort of, I mean, we in the morning we saw some COVID-19 studies from patients as well as from non-human primates and so forth, and where things were correlating. But would that help in terms of, are they good models in model systems like mice or rats, or for lung diseases or gut diseases that to correlate well, or have they helped anyway?
From our point of view, again, I think they can be helpful. For example, we try to compare responses to hyperoxia in the mouse to our patients on ventilators, and we can ventilate, the mice for some period of time. As y'all know, the immune system is not the same. The responses are not quite the same, but again, it's a place to start to look for similarities.
Okay. How about any other thoughts of other folks who are on the call regarding challenges for quality control that we should address across consortia?
I was thinking about this, was very interested in the spike-ins, what the consortia, there's different ways to spike in things like cells or specific RNAs for the single cell. I have the idea to mix something with male and female tissue at the same time, so that most of the cells, for example, for female could be easily identified with specific gene expression like cyst. Something that I would like to hear what thoughts are there and whether this can be followed up and recommended by the consortium or by different groups. What's the best thing to spike in something to get ideas about ambient RNA contamination and so on.
Yeah. So yeah, thanks, Edgar, for that comment. Yeah. So could you elaborate more, like how would you use the male-female mixing and for the quality?
Yeah, there's always, for single cell, there's always the problem to find out who is who. So we know about that you could use single SNPs for every cell to identify. You could even pool different samples. So this would be one way to approach that, to do more than one sample at once, and then see what comes from which sample. But that's I think the other idea is to put in mouse another beast, which I don't like that much, or put in a cell culture system to differentiate. So if you spike in 5% of an animal or from another sample, from another tissue. So I think there are different ways people has done that. And so I would hear from the consortium or from different groups, what's the best thing to do and what's the easiest one. My idea about the two different genders is that you can easily identify bioinformatically which cell, whether the cell comes from the female or from the male. Then you don't have to look at the SNP base, the bases on the SNPs.
Okay. Any thoughts and questions about that? Okay, any other comments about any pre-analytical parameters which we should focus on?
Yeah, my concern with the last idea, well, a good one is it still requires a fair amount of RNA sequencing and processing and pulling apart to do. And if you're not careful, I guess you run the risk of losing your original signal. Is there some easier way to do it? I mean, I know I'm running up against the people doing RNA-seq. Everything is done with RNA-seq. I understand that. I'm thinking more, some indicator dye or
Yeah. I mean, yeah, so is there anything like before going to the whole pipeline, can we use something, in the beginning to assess the quality? Okay.
I guess a question for Stephen Hewitt, if you're listening. To what extent can the pathology be helpful of telling you, or does it just tell you the tissue is really bad, you can't get those intermediate cases?
So, Rob, I am listening. I'm working on some in the background. The pathology generally can tell you the DNA is good and oftentimes inform that the protein is good. But once you want to look at phosphoproteome and RNA, the current pathology in most clinical settings or with most tools is just not adequate to refine that. Now, a couple organs, I think the pathology is probably more telling. If you're in the kidney, the histopathology is pretty telling pretty quickly. But lymph nodes, not telling whatsoever. Livers, they can be pretty subtle. It always gets back to something no one wants to hear me prattle on about, which is, fit for purpose models. And that is, how good does it need to be to accomplish the goal you've got? And unfortunately, and I mean it unfortunate from the pathology perspective, not from the technology perspective, the RNA-seq really, the pathology just is not leveraged to give you the quality required for RNA-seq. Most RNA-seq is done in frozen tissue, in fact. Now, there's RNA-seq from FFPE, but unfortunately, the good news is the RNA-seq on frozen works pretty good. But all the steps just to get you a good frozen section to look at from the pathology perspective really makes it very less informative. Pathologists hate frozen sections.
Okay, thank you. That's what I thought. Not there.
Yeah.
So what you were suggesting is, for pathology, perhaps we should figure out for which organ systems across different initiatives is being used and is useful for their triaging or for quality control, and for which it's not helpful. Is that a task that is useful for a collaborative effort?
So Sanjay, I guess my reply would be, I think the triage is good. Make sure you have the right tissue and gives you a first quality pass read. Because a lot of times getting the right cell type in there is a lot harder. I think really for a lot of these questions, for these studies, you end up using bioanalytic methods directly targeted at the biomolecule of interest to really qualify the specimen in terms of quality. Now, doing that for phosphoproteome is extremely difficult, but there are some reasonable metrics that are available for both RNA quality and DNA quality. Now, obviously, DNA is really not an issue here either. Proteome, total proteome, I think there are actually a couple methods that can be used. Most of them, oddly enough, came from some older mass spec studies, but again, I think they seem to work.
Okay. Any other thoughts on pathology or whoever else is using pathology? Okay.
This is definitely a problem for our lung also, right? You'll see the best histopathology, the best immunohisto or fluorescent chemistry on an inflated fixed lung, whereas all of our single cell work is going to have to come from frozen, non-inflated, non-fixed. So making those comparisons is going to be, challenged by that.
What happens when you inflate the lung? Why doesn't the single cell work?
So needing to get that tissue processed and stored quickly, right, for the single cell stuff. So I can inflate it with air, but it's like inflating a marshmallow. As soon as you take that out of the cold to try to cut a piece off of it, to get what you're going to need for the single cell work, it starts to warm and deflate. So it is just challenging. I know some have done it a little better with the lung and air inflating, coring, and freezing those. With the pediatric lung, that's challenging just by the size. So if someone has a good protocol that they would recommend, we'd appreciate that too. So is that, I'm not sure I answered your question well. But
yeah, no, but we've worked with lung a little bit, but
yeah.
Yeah,
you know, once you inflate it, it's just not a very well supported tissue. So it doesn't stay cold. You get air bubbles, you know, when you try to then put it in OCT or other freezing media.
I think you summarized that very well. I mean, lung is a uniquely challenging object because you can actually get the fluid in, but then of course it really damages things. And in the contrary, on the other organs, you're really limited by the diffusion. And you know,
Right.
working in human samples, it's not like you're working in an animal where you can actually circulate, fix it into the site to do the task. Many groups have tried to move towards needle-driven biopsy approaches to try and improve these qualities where gradients are smaller, but lung is a great example. It's a nightmare.
Okay. All right, so we discussed the clinical parameters, metadata, the tissue procurement parameters, the metadata minimum needed for that, and the tissue histopathology, on which tissues this is informative and where it's not. What other pressing questions we need for quality control?
Oh. Yeah, how about sample mix-up?
You mean like what Edgar was suggesting, or?
I may have missed it, but just the
male and female he was talking about mixing.
So putting some barcoding into the sample, so afterwards you can know which one you had. Not just for high throughput methods, but to ensure that you can trace the tissue.
So Rob, where do you want to put it into the system? That becomes the challenge on this. I mean, if it's a frozen specimen, you theoretically could literally put a drop of fluid containing a barcode nucleic acid on or around the specimen at the time and before it's frozen, freeze the tissue, and you might be able to make it work. It's got a few other challenges, but it might work. If it's a chemically fixed specimen and it's got downstream analytics, then you got a much bigger problem. It's a unique idea. I mean, the idea of using spike-ins for traceability has been discussed in a number of other platforms. It's real. I've never tried to do it in tissue. I know a couple of people that have tried similar ideas, but they're challenging.
Yeah, I guess I wasn't just thinking DNA, though I don't know how difficult the analytics are. I was thinking more, mass spec or something.
Yeah, I thought about these also quite a bit. So I was thinking to put the genetic barcode into the name of the sample. So if this sample is always for RNA-seq is always at some point there is always the way to identify them. So either with some SNPs, there could be the identification via sequencing the genome, so, or for at least 10 microsatellite markers, or specific amount of SNPs. They could be decoded in a name, in the sample name. So that's, I'm not sure about the legality of that.
What do you mean by legality?
I mean, whether it's legal to put to have file names corresponding to the sample, that it's traceable who it came from. So because that's pretty unique, but you don't need, if you have only say 10 SNPs, then it would be not identifying out of the whole the population, but that would be at least it would be securing that we have the correct sample. So this can be always traced then.
Okay, any other thoughts?
Did we talk about shipping, ensuring integrity of?
Yeah, so briefly in the previous session, we touched on storage and shipping, or for at least folks who are shipping, slides or tissue across. And I think Alexander pointed out that therefore their imaging, they are shipping slides and stuff. So one of the things could be to standardize shipping protocols. Is that what you meant, Rob?
Well, I was thinking more, I mean, I think that's a good one too. The and would be that they're being shipped at the proper temperature. It gets back to just, how do you know, just tracing the tissue and how do you know what really happened to it?
Yeah.
And was it within some tolerance? Again, not perfect, but some tolerance.
More tracking, shipping. Put it in the blue box.
Right.
Okay. So let me just, so that we don't lose track. So the items that we are sort of talking about is so far I see in two categories in the blue sheet. One is sort of related to metadata acquisition, which includes the minimum metadata needed for clinical specimens, tissue procurement, or tissue histopathology, identifying what's informative and what features are helpful. And then the other one is metadata regarding shipping and storage conditions. So that's sort of one theme that is developing. And the second thing which we were talking about is sharing protocols, which will include what methods work across tissues and across consortia, and what does not work. Anything we want to add more to that? In terms of the second theme of sharing protocols, maybe what are the common artifacts?
If I can ask a question that I asked this morning in a different group, but are there things that people try to do in the coronavirus example, which was presented quite beautifully and summarized really nicely? Were things that you tried to do that you think was limited by sample integrity? And then just as a way to reframe this a little bit. And then are there ways around it? I mean, maybe it didn't come up at all, but just wondering.
Yeah, can you elaborate that one, Rob? I sort of missed it. What?
Yeah, so I'm taking the example this morning of the coronavirus, which was a beautiful example, and a lot came out of that. But my question is, in the process of, if people here were involved in the various analytic pieces of it, were there times where you thought you were looking at artifact but weren't sure? That's in other words, where are you bumping up against what might be artifact? And is there something we can bake into the systems
So how to recognize an
that make it easier to figure it out? Does that make sense?
So how to recognize an artifact?
Yeah. Well, part of it is that,
Mm-hmm.
and then maybe how to trace it back.
Yeah, so how do we really know if we don't know the ground truth, how would we know what is really an artifact and able to trace it, right? I mean, we don't, because we don't have enough data, right? So,
Yeah, but if you're looking at, 10 or 20 lung samples and one of them looks clearly different.
Right.
Just for example.
Right, yeah. Yeah, and we see that in kidney, that we will see one patient who has a particular cell type or clusters that, not seen in others. So then the question becomes, is that an artifact due to how long they were on ventilator, or is it really true biological variation? So
Rob, I think you're kind of asking what are the factors that cause those artifacts, right? Can we identify them?
Right. Yes.
And therefore avoid them, or at least be aware of them.
Yes, exactly.
Yeah.
And Rob, I'm sorry, I cut you off. What I was going to say is sometimes my approach has been, since I don't know what those factors are, to go ahead and collect more data and then start to tease it apart on the backside. Now, that means I have to have the metadata to help me. But even then, even without specific metadata, if I can put them into groups, it moves me one step further.
Okay.
So should we put that in the first category or the second category? Should we put that in the metadata category or in sharing protocols category?
Yeah, it sounds like it may be some of both.
Okay. Okay.
Yeah, if it's an artifact, we could, with reproducibility, we could figure that out. If we can do it on a same sample kind, repeat it, and then the artifact should go away if it's technical. That's one way to do it. Or do bulk RNA sequencing and deconvolute later and see whether this is keeps on.
Okay. So I put in how do we trace sources of artifacts, how do we recognize artifacts, and then what are the common genes associated with artifacts across consortia. Anything else we should pose here that I missed? Okay. What else? Is there anything else that we should prioritize before we start focusing on how to do items in the blue final template? Anything other pressing or stumbling block that people are facing when they are analyzing the data and they wish that, oh, this is a critical part of QC, we wish we should have gotten or obtained, or it's challenging. You know, I mean, this is the time to brainstorm those if we have not already discussed.
You know, so much of this comes down to communication.
Okay.
And how in our limited hours of the day, right, can we maintain the communication between the consortia? Again, none of us will deal with enough probably to be able to answer these questions by ourselves.
Right.
So that would be my plea to those that are creating the databases or handling the communication, that there be some way to list, or, you know, have a running, I don't know the best way to do it, right? I don't have enough time to read what I have so far, so. Anyone have a suggestion? Communication of experience.
Evelyn Wood, reading dynamics.
Say it again, I'm sorry.
Evelyn Wood, reading dynamics.
Yeah.
If you're older than 50.
Okay, so let's discuss about like recommendations or like what we want to do. We have identified these questions. So how are we going to go? So let's say for the first part of the metadata acquisition. So what are our recommendations of what we want to do with this? So what would folks propose? We solve the idea of the metadata collections. What is the regimen, you know, for folks who want to read this and say, okay, Gloria mentioned communications. So the communicators need to communicate this to the consortia. So what is a recommendation for minimum metadata needed for clinical parameters and for tissue procurement parameters? Do we say people collect their EHRs or something like that? You know, so this is, I think, we need some sort of an action item, you know, like how we can go about achieving these tasks. Yeah.
I don't know, just to,
Go ahead.
go ahead.
Yeah, go ahead, Gloria.
So just a suggestion that I think this is organ dependent. And one of the places to start might be to get those doing the same organs or with expertise in the same organs to compare their metadata lists and maybe have a conversation about what is helpful, useful in an analysis, what might be in the future as we get more data, you know, and what is just not feasible to collect.
Yeah, and I think that's an excellent point here. You know, that's a beginning place because we do have across HubMap, HCA, Chan Zuckerberg, and LungMap, and so forth, many folks doing, you know, same organ. And I think it's a this is a pretty appropriate forum to start making and comparing these lists. You know, and I think this can be applicable to all the problems that we are identifying here. The clinical metadata, the tissue procurement data, you know, and you know, all even the histopathology and so forth. So I think that's, I mean, I would agree with certainly with that, that we should start recommending at least as a low-hanging fruit to do that. Yeah. What are the thoughts of some of the other folks?
Yeah, I agree that with Gloria that this will vary according to the organs that each consortium is studying. But I'm wondering if there is any way to articulate with CZI and Hamsley to get this sort of information compiled to the general public, for instance.
Yeah, I think we would need, I mean, this probably may have to go to maybe higher level organizers who organize these efforts and then maybe, you know, collect, you know, these like maybe working groups, you know, to collect this information. And, you know, we sort of started doing that with HubMap, where Ajay was collecting metadata elements from all the organs and from people who are doing kidneys and trying to, you know, merge them. But it was a Herculean task. It wasn't easy. It was very difficult because terminologies are different, and what everybody collects or gets from their clinics or from organ procurement sites is very different. And to get that in a de-identified manner is another challenge and required policy issues and so forth. So it's not an easy task. But for if we want to have track quality of what we're getting, I think it's something is needed. Yeah. Yeah. Okay, so how would we make prioritization of must-haves and could-have? Okay, what else? How else should we? What else should we recommend uh, for other aspects that we discussed in metadata? Sharing of maybe similar tissue across different, I guess, sites. Would that help into refine?
That would quickly figure out whether they're systematic artifacts. Yes, that's a great idea.
Okay.
The other thing I was going to add to the metadata part is probably already is just I mean, the hard part here is when you don't know exactly what's causing harm to your tissue, what do you put into that metadata? And, you know, like in KPMP, do you take a video of the whole process?
Yeah.
And that becomes the metadata, or all the timing information, or the nurse with the iPad taking pictures of things and recording time automatically. I think initially a lot of that has to happen. You probably need to over collect for a little while until you get some idea of what's needed.
So Rob, this is where KPMP has had an advantage, and that is you have multiple sites collecting the same organ. For these studies, it's a little bit more challenging because every site is collecting generally a different organ, or you might have two sites at the same organ. And for some of these organs, the restrictions on what you can get are so complex, unfortunately, as you take what you can get. For others, you could work a little bit more, but it certainly makes debugging this a little bit more difficult, because it is not a situation of what is good for the goose is good for the gander.
Yeah, yes, I agree. But now let's turn that around and ask, okay, given that these things are being collected in different ways, and yes, they're different organs, I get it, but is there a way of deconvoluting out a tissue processing signature? But it's just impossible.
So what's your recommendation, Rob?
That was a question, not not a recommendation. Okay, well, we keep going. Let's get back to less theoretical, more practical issues.
Okay. Okay, so what we have, just to summarize so far, recommendations for comparing lists, then document extensively how tissue is collected, and then sharing the same tissue across sites to check robustness of QC measures across technologies. So this hopefully should cover some aspects of artifacts, as well as these issues with the procurement parameters. Any other recommendations on how to do this?
Yeah, I thought about that we always do bulk RNA sequencing for some of the RNAs, for the single cell RNA sequencing as a comparison. That the same tissue is always also sequenced by bulk to have a standard, which is one of the, which is then a reference.
So that helps you to find what, like what you're missing, and how.
Yeah, for single cell, we might find that we miss some cells. Yeah, there might be a bias by the dissociation process. Of course, we know that there might be some cell types maybe missing, or the proportions are also probably because of susceptibility of the tissue. Some cell types might be different, and bulk sequencing and deconvolution might help to find that out. So that could be one way. There might be others.
Okay.
Yeah, this is a valid point, yeah, indeed. What we've been doing as well to benchmark because we're planning to run mostly single cell RNA sequencing on intact cells, but to benchmark the isolation protocols, we are running from exactly the same sample, some single nuclear RNA sequencing as well. So yeah, bulk RNA sequencing, single nuclear RNA sequencing are, I would say, nice strategies to really benchmark the isolation protocols and to keep track of things, at least in the beginning, to be sure that we're not missing a cell type or cell state.
Yeah, I mean, for us, for single nucleus, we did not really find, at least for us, much value for bulk anyway, because for our single nucleus atlas, so we don't really do that routinely. In the beginning, we did it, but then we don't do that anymore. Just go because we have some starting reference, and we just benchmark with that, and the quality, across the different technologies as a benchmark.
Yeah, in the beginning it could be more interesting, but later on, yeah, bulk RNA sequencing could be more appropriate to have as a reference. Yeah.
Yeah. Once you have your whatever,
Mhm.
you can go back to bulk, but initially
Mhm.
the single cell. Yeah.
Yeah, indeed.
I mean, so those I think 3 activities are for this first part of the metadata acquisition are something which I think are feasible and practical. How about communication? Gloria, you mentioned communication. So any recommendation about communication efforts, how it can be done consortia-wide?
I'm going to apologize. First of all, I was typing away there with my mic on. I hope that wasn't too annoying.
Oh.
Well, I think what we're doing right now is a good step ahead in terms of communication. I think encouraging our various DCCs, or whatever you want to call them, that are handling the products of each of the consortia, that they put thought to kind of building a database of experience and building a database of these sorts of issues that, again, won't be the analysis that gets published. Although some of these things that we're talking about, maybe we should think about publishing them. Or, as Alex said, making them available to the general public, not just to the consortium members. Yeah. So encouraging each of the consortia to put some time and effort into distributing this kind of experience and expertise that comes from the work. Yeah. Yeah, I'm hopeful that this is not the only gathering of the various consortia. I don't think it will be. Many of us are involved in more than one, so that helps. But yep.
Okay. Anything to add to this first blue point? And we can go to the second one, blue set. Yeah. Okay.
Well written.
Yeah. So for the sharing of the protocols, so we have the problems of what methods work across tissue and across consortia, and what does not work, and what are the common genes associated with artifacts across consortia. So recommendations for this, of how to do this across, the different sites and consortia. So how would we work across consortia to, share this knowledge, regarding what works or not? I know several of them encourage having their protocols on protocols.io, but so a lot of these protocols are available, but the coming togetherness in this fashion, that the point that Gloria raised, is sort of missing. So how do we make sure we tie these together? I mean, if you have any recommendation or any innovative ideas regarding this? People maybe who have a very big picture from the, higher up, who maybe may have some ideas as well, how to do this. As a task force.
I don't have an answer for this, because this just isn't my space as to how to pull the consortia together. I think you've hit an important topic, but at some point I just don't know exactly the boundaries in which I can play with across this one.
Rob, do you have any insight on this? Anyone else who have any ideas of what could be a good viable way so that we can get this information so that everybody can benefit?
To be honest, I'm not sure if this is something that we can really tackle systematically, perhaps having working groups exchanging in an open way, or even protocols.io, yeah, some sort of platform giving space to work groups. I think this would be the best alternative.
I think that's a good suggestion. Working group this morning, the similar question came up about, why hasn't this moved forward and everything? And part of the problem is that you didn't have enough people to have enough energy to overcome the entropy issues. And so we have to get the consortiums talking together so that they will interact and move forward. But again, it also needs deliverables that investigators can feel that they're getting their credit for, which is protocols on IO, manuscripts, white papers, whatever it is. It's an odd situation. I'm going to be very honest. It's kind of an odd situation because in some ways, the COVID-19 shutdown across the research labs is actually giving people an opportunity to actually have more discussion and think about writing these documents and work together electronically rather than just writing, constantly writing the papers and collecting data. No one can collect data right now. So you either can analyze data or work on doing the rest of this. And so this may actually give us some breathing room to put our heads together and make some progress.
Yeah, that's a good point. Okay, so I think, everybody can hopefully see. Is there anything else missing in terms of recommendations? I think we've brainstormed.
This might be a little bit lame, but I'm going to say it anyway. Part of it is just the education of especially of the people doing the analysis, to keep an eye out for those outliers and what they might represent. To keep an eye out for the artifacts that may be biological or may be due to our processing. So just raising that awareness and keeping it as a valuable outcome of the analyses that get done.
Hi, this is Rob. I lost my earphone things. The battery died. I'm back again. This looks like a quite good list. Let me see what's missing.
So I'm going to put report outliers in the sharing protocols, right?
Yeah. Do you have sharing protocols? Is that here?
Yeah. Report outliers.
So I have a question. How effective are the protocol sharing websites for these projects? Are people really using them? I'm kind of blind to it, so I'm curious. What do you think about these platforms? Are these working for us? And if they're not, why not?
So, that's a great question. So we have some protocols in protocols.io from HubMap and KPMP. The only way I can see how they're used is I sometimes get a notification or I can see how many people have viewed my protocol. That's the extent to at least what I can tell, if how much people are accessing. Whether they're being used for collaborations or interactions, I don't know. Maybe others may have more experience. But,
So from my experience, I've been going there especially to get insights or sometimes to double check on things I'm doing. I would say that the hot topics or the protocols that are hot in the moment, there is a very active exchange around them. And then some other protocols that are not that popular, of of course, they are laying there without that much interaction. So in general, I would say for the protocols that are really that have a lot of interest around it, I think it's quite active. But in general, I also have the point of view that in many cases, the protocols that are there are from studies that are already published. So sometimes once they are made available, it's a bit too late. But this is something that is difficult to enforce as well, how to get protocols there in earlier stages once they have been benchmarked. I think this is a challenge that is difficult to be tackled. But other than that, the hot protocols, I think it's quite active, the exchange.
Does the commercial nature of it affect anyone?
Yeah, you know, when we were, I'm sure when we had these discussions in KPMB when we were initially, you know, pondering on this, and I looked at all the different small print of protocols.io, you know, but then ultimately we decided because the value gained was a lot more, and many other consortia were using it. There was a commitment of you know, safeguarding the protocols in case the commercial entity was you know, goes under or something. You know. So at least I think that was the motivation. So. Yeah. Okay. So any expertise we need, additional expertise we need for some of the challenges which we have encountered, or this is something we can do, you know, after, you know, this, is there any emergent additional expertise we need for these quality control measures? Anybody can think of whether it's metadata, I mean, should I list communication or organizer for these efforts as an expertise? Somebody who knows how to coordinate all these different, you know, working all different consortia together. Is that, you know, something key, I think, looks like to me. Otherwise, everybody will be siloed, you know, if we don't have a moving force, right?
Yeah, I agree. That's a good point as well. Yeah.
And I'd say it's organizers, not just like us, but sorry. Yeah, so beyond us, the small, relatively small group that we have here, and also the expertise in the communication tools, who can actually, you know, take ideas and make them into a functioning tool.
Okay, I'm gonna copy and paste this for both. Okay, all right, so anybody want to volunteer their email contacts? I mean, they will have the folks who are joined in the chat room. I'm happy to put in mine. You want to, Gloria, can I put yours for the sharing the protocols one?
Sure.
Okay. You go by G. Pryhuber?
So my email.
Okay.
Is that what you want? Gee, I'll put it in. It's easier.
Yeah, that's great. Thanks.
Just easier. Yeah.
Okay. If you know, if there's nothing else, I want to thank you very much.
Sanjay, can I suggest that everyone, the 12 of us or so that were listening in here, that we each put our names under that other group members, so that we can get back in touch with each other and just please know who's been in the conversation.
Yeah, please feel free to add on the Google Doc your, you know, as an active participant, you know, so you can contribute to some of these challenges. And thank you very much for joining in and sticking this. and brainstorming on these issues.